Hello everyone, welcome to Stat 479, lecture 24. So this is going to be the final lecture for this course, and it'll be the last of the four lectures where I give you some kind of hints and some basic frameworks and guidance on what's kind of expected for the project. In some particular, remember that there's 9 tasks, and today we'll focus on kind of tasks 7, 8, and 9. In particular, I'll first talk a little bit about how to think about 7, 8, and 9 through two different kind of criteria, which are prediction and in interpretation, and I'll talk about what those are. Then I'll focus on task 7, which is really about validation, in particular, a couple of different types of validation, like prediction, stability, and domain knowledge. Then I'll talk about the 8th task, which is about connecting back to the scientific problem or the scientific question that you started with. And then I'll talk about part 9, which where you need to kind of think about what you would potentially do in the future based on what you've done in the project. And we'll again connect, and I'll kind of talk through all of these in the context of the example that that we've been sort of focusing on for these 4 lectures. So that biochemistry example where we had 988 samples, where you have sequences with 8 different kind of positions and different parents in each of those positions. So just to kind of recap what we've done sort of so far in the context of what we've covered for the project so far. So there's 9 tasks overall, and broadly speaking, in the first lecture, we focused mainly on tasks 1 and 2, which is really just focusing on the scientific question, understanding kind of where the data's come from, so the data collection, because where the data comes from really shapes, it really shapes what we want to do with the data. And then the third and the fourth step, which really focus on exploratory data analysis. So just doing some very basic checks of the data to sort of understand, like anything you thought about the data collection, you can try to verify, and you can do some basic sort of types of simple analyses to see what you might expect without doing anything too complicated. So taking things like averages, distributions of the covariates and that kind of thing. So just very simple sorts of analysis. And then also think about the data pre-processing. The exploratory data analysis will tell you a fair bit about the data, and that's going to be really useful when you think about data, like how to pre-process the data. Do you want to normalize the data, as we've seen in the example here? Here we have categorical variables in the x's, and so we need to kind of use encode those through the factor function in R, or the one-hot encoding, which I described. Then in the previous lecture, in lecture 3, we really focused on kind of the implementation of the methods that we considered. So we thought about what methods would be appropriate. So we for the example that we considered, the GLM was reasonably appropriate and GLMnet as R packages. And then as I mentioned throughout, you need to also think about sort of validation and this notion of training error and test error and so on. Then we can in that context you can we talked about dividing the data up into training, test, and validation sets, and in particular we spoke about this notion of k-fold cross validation, which is a very good way to sort of validate your various different methods, and it also gives a way to potentially select any tuning parameters that you need to select for your method, like for the lasso or the ridge, where you need to select lambda. So today we're really going to focus on 7, 8, and 9, and this is really broadly speaking going to say, well, you've run your algorithms and you've got kind of your outputs of those algorithms. Like, what do we do with that? Like, how do we interpret those outputs? And that's going to be kind of the focus of the lecture today. Okay, so let's begin with and one thing to note here is that 7's really in both parts here, the validation, and that's kind of important because the way you implement your algorithms is going to be tied to how you want to validate at the end, which is why I spoke about it in the previous lecture. But then this part of, I guess, the validation really goes into more depth about, what you can actually do to kind of guarantee validate, like, what can you do to actually validate your data and actually show that whatever conclusions you come up with kind of makes some intuitive sense. So that's why kind of 7 appears in, like, that 7th task is really important, and it kind of ties into both the model implementation, but then also how you want to interpret and present your results at the end. So let's talk about validating results. So I guess before going on, I'll just talk about so I'll say that, well, I guess I should say, so there's often so this is broadly speaking, 2 main goals. When developing so the first one, which we've already touched on, is prediction. And what that just really means is, do we fit our data well? So when we say fit data well, we're typically talking about something like the test errors. So, and this is usually a basic requirement of algorithms. So what well actually means, I mean, it depends on the application, because as we saw for the for the biochem exam, we could actually fit that data reasonably well. Like, we got an accuracy of about 83-84% just with simple logistic regression with main effects terms. But for some other applications where the data is more noisy, if you're looking at, say, like finance data or more sort of social science types of data, fitting the data well may be like an accuracy or a correlation or an R-squared score, depending on how you measure it, is something like 60% or even 30% might be a very good R-squared score. And so the first question is, do we fit data well? And so well is a relative term here, and it's relative to what other algorithms do, what state of the art algorithms do, and that's sort of really just saying can you is your does your model kind of fit whatever data you have, so both on the training data and on the test data. The second thing, which is kind of, I would say, more interesting but also more challenging, is this question of interpretation. And this really is, there's very broad definitions of this, so I'm not going to kind of be too broad and focus on all aspects of this. But this is really looking at things like, variable selection, which variables are relevant. Okay, so I didn't say. So this is like variable or model selection, which is saying, which variables are relevant. So I sort of came up with that list of those 6 variables from the previous lecture, and that's also output in the code from the previous lecture. That's really like not, you're not necessarily asking which whether you predict the data well. You're just saying, can you tell me which of the parents in which positions are going to influence functionality. There's this other thing that's very important, which is that a lot of people care about, which is causal inference. That's quite a hard question. So I'm putting a question mark there, and and this is really saying, can we draw any causal conclusions from the output of the algorithms. And this is also and another thing that this sort of ties to is they're just data-driven decisions. What decisions can we make based on outputs of algorithms? Okay, and so this is like really sort of the heart of what we're trying to do. So interpretations, what we typically care more about. So predictions, really like more of a good sort of sanity check to make sure that that our model is fitting things well. But what we often really care about in many applications is which variables are relevant. So what and that can influence infer things like can we draw a causal conclusion, like can we say that if we in the example that we've spoken about here, if I put this parent in this position, that's going to increase functionality. Is that a causal conclusion, or are we just observing some kind of correlation? Or if we're looking at voting data, is there a causal connection between gender and who someone's going to vote for, or is it is it just a correlation or association with some confounding or something else? And so those are all kinds of very important questions to think about. And I should also mention that this is actually it's often very hard to make a causal conclusion. It's usually dependent on where the like how the data was collected or what the experimental design is, and I'll talk a little bit about that in the context of the application we've studied so far. And then finally, what sort of decisions can we make out of this? So often when a practitioner asks you to implement some sort of data analysis or to do some inference, they don't really they aren't just doing it to see if it predicts well. They want to make a decision. So for example, they might make a decision in the context of voting is, okay, well, should we focus on this demographic of people more or this state more in the context of the election? That's something certainly people both parties did a lot when they're thinking about where do we want to focus our attention. So that's a data-driven decision. In the context of this biochemistry example here, we're looking at, well, how would we design a better protein to kind of improve our functionality? And so that again is a data-driven decision that can be made. And so that's that's sort of leading to these data-driven decisions. So I just want you to, whenever you're thinking about validation or presenting the results that you have, think about both the prediction, so how well does it fit the data, and that's what I spent most of the previous lecture talking about, but then also the interpretation. So from a scientific point of view, what can we say about what we've concluded? Okay, and so that applies to all of these last three steps, 7, 8, and 9. So this prediction and interpretation apply to 7, 8, and 9. Okay, so now let's focus on so if we want to focus on validation of methods, so this is the seventh the seventh task of those tasks listed for you to do. So for the validation, there's, I would say, broadly speaking, there are three types of validation. So the first one is prediction performance. And that's that's really talking about the test or validation set error. Okay, so one thing I should also mention is that you should really only use your validation data, which you haven't really touched during the training process at all, to do your validations, because otherwise, otherwise you're, as I mentioned, sort of overfitting and seeing the data. So the prediction performance, this is a test and or validation setter. And one thing to mention here is that the choice of error metric is very important here. Very important here. So for continuous data, often use mean squared error. But this isn't, and we've often used this in class as well in the first 20 lectures when we're trying to compare different methods. But this isn't necessarily the only or the best choice. For example, if we want to be more robust, often use L1 error instead, because that's kind of more robust to outliers, and that's what leads to kind of using things like the medians or the interquartile ranges rather than means and variances. For classification, we simply look at types of errors made. And so this is, we even though the output of the algorithm might be some number between 0 and 1, we, as I saw in the previous, as you saw in the previous lecture, we thresholded it at 0.5 so that we can see, what sort of type 1 and type 2 errors do we get out of it. And I should also mention that in general, likelihood, and then this other notion of deviance, which if you look at the output of a GLM, they'll often report the deviance, is a widely used measure that is tied to the model. So that's one thing to keep in mind, that one of the things that is both good and bad about things like likelihood loss or deviance, is that these kind of apply to any statistical method. But the other thing to keep in mind is that they're very tied to the method, because you're kind of looking at the deviance or the likelihood using that particular method, whereas things like mean squared error or type 1 and type 2 errors for classification, they kind of apply to, they don't really bias your results to any particular method. And then the other thing that you might have is if you want to select a graph, or I should say if you want to learn a graph or a network, you might need to think about other metrics. So for example, you might just use the simple thing that if you get the graph wrong, then that's a fail, and if you get it right, that's a success. But that's a pretty harsh criteria, because you might get all of the edges right except for one, and so instead you might look at something that looks at how many of the edges did you get correctly, or something like that. But what I'm saying here is that you have to really think about what error metric you use. And as I've always said, there's not necessarily one right answer here. You just need to make a decision and justify that decision in your report. So how do you choose that error metric? So you don't necessarily need to blindly choose like mean squared error, even though that may be the best choice, or if you're doing classification, just a particular error. Just you can think about different types of error metrics that make sense for your particular application for your particular data, and justify that. So that's kind of the first thing to keep in mind, and that's so prediction is really the first type of validation. So what we're really saying is that the method's really only considered a good method if it predicts well. So better methods should, in general, have better predictions. Or I should say better methods or models, maybe. Should have better predictions. Okay, so that's the first thing about predictions. So just to kind of say, for example, in Biochem demo example, in the Biochem demo example, we had test error of about, I think it was 80, so it was or test error about 16.5% roughly, which is pretty good. That's a lot better than random guessing. But we will usually have many different methods that we compare. So as I mentioned, I just ran logistic regression with main effects model, but there's also, as I mentioned, logistic regression with pairwise interactions and main effects, or things like the lasso or ridge regression, and all of these are going to get different test set errors, and so this is kind of like a way to compare methods as well using prediction, and in this case it would be like misclassification error. The second, which is really important and often maybe not sort of thought about enough, is this notion of stability. And that's sort of really saying like how stable are our conclusions, both prediction and interpretation. Okay, and that's really important, because when you report results, you don't want it to be that it's so sensitive to if you change the data a little bit, because remember, this data is collected some way, and if you'd had a slightly different data collection, if you'd had the same data collection process, due to randomness, you may have got slightly different data out of it, and you don't want to that your conclusions to be highly dependent on the random draw that you picked, or if you just tweaked the data collection a little bit, like you wouldn't want the results to change dramatically, because that's sort of not very reliable. And so another way to think about this is if we perturb the data or algorithm slightly, will same conclusions hold. Okay, and so that this is really, really important. So So a good check, which I did touch on briefly in the previous lecture, is if you do k-fold cross-validation, are the k models similar or different? And that's kind of a first good check. Because if you're doing this k-fold cross validation, your models are changing a lot. That's telling you that there is not, that there's not, that your performance is that your approach is highly sensitive. So to measure similarity, there are many choices, and so this again ties to what's your goal. Are you thinking about prediction? Are you thinking about interpretation or causal inference? So one might be prediction error. Again, that always kinds of comes up. Like, does your prediction error change a lot if you perturb the data a little bit? But then you can start to think about interpretation. Do variable selected chains, selected or parameter estimates. Change drastically. Okay, and so, for example, we found 6 highly significant variables using the GLM. If we take half that data, would those 6 variables still be significant? Okay, and so that's that's kind of tying back to interpretation. So that's another sort of performance metric you'd use. And there's a lot of choices here for how to compare variables. You could look at the set of variables for the different k-folds and see if they're identical. Like if you took maybe the top 6 variables, would they always be the same or would they be different? But that's another kind of measure of stability. And that's that's sort of really really important here. And this is also can you find so the notion of stability is also very connected to the notion of variance. And so can you find confidence intervals? This is this is quite challenging. For simulated data, the way you can typically do that is to generate like hundreds of different data sets. So you can generate like 100 different data sets and maybe just kind of like apply sort of normality types of assumptions to these. So you can sort of like often make normal approximation normal approximations. That sort of helps if you want to derive confidence intervals. And this is not something you have to do, but something to think about whether you can actually come up with error bars. Like practitioners love error bars. That if you can come up with those, and sometimes it is certainly possible to do that. And then there's other methods like you can use, and I won't go into too much depth on this. If you want to, it's certainly something you can use. Like methods to generate multiple data sets, e.g. So e.g. The as I mentioned, cross-validation, or what's called something that's also used in statistics, which I haven't spoken about, or bootstrap, to come up with confidence intervals, and that's sort of the another way to measure stability. But there's lots and lots of different ways. Another thing that I should mention is that algorithm or method perturbations. So what I mean by that is, if, for instance, you change a tuning parameter slightly, will you dramatically change results? Dramatically change fitted parameters or prediction. Okay, so data perturbations are really when you kind of sample the data and you get a slightly different data set. It's very similar but slightly different. Does that sort of massively change things? And the other one that you can think about is algorithm or method perturbations. So, if you change lambda for lasso a little bit, so this is a slight. If you change lambda for the lasso slightly, does that significantly change results? So I just wanted to mention that there's in addition to as I mentioned to you earlier, there's a lot of design choices that you actually make as you go along, and a lot of these there's no kind of clear right answer. This is a choice you made, and so if you change that choice that you made in terms of designing your algorithms, will that dramatically change the results? Because if it does, then that's also a bit of a red flag that you're very sensitive to sort of model choices you make, and your conclusions are not overly valid. But if you are somewhat if you are robust or stable under these perturbations, if you made a different design choice and got the same conclusion, then there's more sort of evidence that you can kind of trust the results that you have. Okay, so that's all I'll say about stability. Again, all of these are very open-ended, so you could kind of talk about these issues for you could have whole courses that discuss these issues, but I'm just sort of scraping the surface on these, and I expect in the project for you to just address these issues to sort of the best to the ability and to the best ability that you can, given that the how much given sort of the time constraints and what you can kind of put into the project. So I just kind of expect you to be able to think about and address these issues in your report. And then the final sort of sorry for this numbering. So the final type of validation is so domain or scientific knowledge. And this is often hard. It's this is something that isn't directly in the data. It's something that is there external source or information that supports the conclusion And this can be in the form of like, for example, a different study or data set, a different study or data set, um, uh, comments, or I'd say, I should maybe say, expert knowledge in the field. Expert knowledge in the field, and the other one is just kind of common sense. So, for example, if you're thinking about like the impact of various different demographics on voting, I guess if you find in your model that, like, say race is not a significant variable, that's probably telling you you're doing something that the model is telling you something that's not very sensible, because certainly there's strong evidence in a lot of other studies, and it's kind of common knowledge that race does have a big like influence on voting patterns. If you find state is not a big influence on voting patterns, then that's probably telling you again that there's something odd about your model, because that again is widely sort of understood to be a significant influence on the on the voting patterns. So this is often, depending on how complicated the example that you have is, this might be difficult to get. So for example, in my sort of demo example, one thing that one conclusion is that linear model, or I should say, rather than say linear, I'll just say, is that the GLM with main effects fits model well. And this is actually something that this is sort of backed up by science, from the perspective that and this is something that I've discussed with many, I guess, biochemists as well, is that in some sense, if you like what the linear model like what the GLM with main effects, if that is a good fit, what that's basically telling you is that if you make two changes, then effect is similar to making one change at a time. And so what that kind of means is that what that means is that if I change x1 to 3 and x2 to 2, if both changes individually improve functionality, then if you put them together, they are likely to jointly improve functionality. Okay, so, and then again, there's sort of science to kind of back this up, because like usually like an improvement in functionality is due to sort of a very local change in the amino acid sequence. And so if you make a local change in one part of your sequence and then make a local change in a different part, they're collectively likely to improve the overall functionality. And this isn't true for everything, but this is sort of a broad conclusion that we can draw. And then you can also, so this is sort of backed up by some sort of scientific I guess knowledge, domain knowledge, or common sense, or whatever you want to call it. And then the other thing you can do is for the six variables that are highly significant. You can discuss with the practitioner. So again, this is not necessarily always available to you, but one thing I expect to see kind of in your report is just at least a discussion of, if we think about this, forget about the data and everything, just think broadly about the conclusion that we're drawing. Like, does it pass the basic common sense gut check? And if it doesn't, then that's potentially telling you that something weird is going on that you need to think about. And again, the more you know about the data, the more you can say about this. So this sort of goes back to that first task of choosing the data set. You want to kind of choose it in a way that the more you know about this, the better. And again, you might be in a scenario where you really can't say anything about this, and that's okay to put down. Just being honest and saying that I don't really know enough about the domain or the data set is that's like, it's enough to to make a conclusion about like this. That's that's okay to say. It's better to do that than to try to kind of talk your way through it. Okay, so we spent a while on validation because it's a pretty important topic in all of this. The next thing is this: we want to connect to scientific question, and so this is the that eighth task. So remember, and this is so again, we're now sort of forgetting about the data. We're thinking more about the conclusions we've drawn. So go back to original scientific question. And so in this particular example, how does a protein sequence influence functionality? And so what we can say from the scientific, if we want to address that question scientifically, we can say that we found six significant or I should say six highly significant sequence sequence positions, and those are listed on the previous in the previous lecture, and then also in the R code. And the other conclusion that we can draw is that the GLM, or I'll say logistic regression with main effects provides good functionality prediction. So what we can kind of say there is in This kind of connects to what I just said about, it means that because these effects are kind of additive, it's basically saying that we can design better sequences, or when I say better, I mean more functional sequences. Sequences by putting together parents in positions with positive coefficients. And this is another really important point. Sometimes the most significant variables actually correspond to like negative coefficients, and so interpreting the difference between positive and negative coefficients is really important. So a negative coefficient here means that if that change is made, so if that parent appears in that position, you're actually likely to decrease functionality. Whereas a positive coefficient means that you're likely to increase. So you really want to be picking the changes that are going to make positive changes, not the changes that are going to make negative changes. So here's just a couple of conclusions we can draw that kind of connects back to the scientific question that we have in mind. So again, this is sort of a very important thing in kind of wrapping things up and making sure that we're connecting back to the scientific question that we started with. So I'll again, and this depends a lot on the actual question that you have in mind. So I've kind of gone through what I might say for this particular example, but for your particular data set and scientific question, the types of conclusions you can draw and what statements you can make are going to change. And so that's just something to keep in mind. And then the final part, which is the future directions or next steps. So this is that ninth task that's listed. So the ninth and final task. So once again, you can say you can divide these into different things. So future directions or next steps, they could be more or different algorithms to try. Another one could be generating different or better data. So you started with data that you used for this particular for this particular project, and you implemented various methods on it, but you might have found that, okay, based on what the study that we did, we found that maybe there's different types of data that you could generate that would actually tell us a little bit more. And then, the other thing could be sort of next scientific hypotheses or questions. Okay, so again, so I just want you to kind of address all of these 3 points when you're talking about future directions. So just in the demo example here, I'll just talk about how I might try to address those. Again, I'm gonna kind of go through it quickly because we only have about 5 or 10 minutes left. For different algorithms, you might think about, as I said, incorporating interactions, that's kind of one key question to think about. Then using lessor or other regularization, and as I said, these were things that I could have kind of done during the project, but I didn't. But they could be viewed as future things to do that we didn't really try. Other things that again are, you can go beyond the scope of the course potentially. Like you'd say, try other methods like random forests or deep neural networks. So you can certainly, you can feel free to be, as liberal like as you want, with this, because there's obviously many different types of approaches out there, and you want to kind of like show that you kind of understand that what we've sort of learned in the course. There's potentially other sort of better methods out there. Okay, and so the other thing that I would also potentially suggest is that we could potentially use is that we could use domain knowledge about specific amino acids and their chemical properties and their biochemical properties. So this was something, we treated each kind of amino acid as essentially the same, and we broke these into different categories based on the parents. But, there's various chemical properties of an A or a G or a T or, a V or a amino acid. And, that actually, this is something that you can potentially look up. And that's something that if you incorporate it into your model, you'd potentially get much better predictions and maybe a better understanding of what's going on. So incorporating that sort of domain knowledge in there would be great. In terms of generating different or better data, we cut up proteins into 8, subsequences. And then the question is, can we make finer changes? So, for example, just substitute a single amino acid and determine the change in functionality. So this is really kind of getting to the question of the data was generated in a very specific way, but is there a way that we could kind of explore the protein space in a different way by looking at different ways to generate the data? So we want to kind of explore this whole protein sequence space in a very different way, and so that that's a sort of a future question that fits under the generating different or better data to kind of get a better sense of what's going on. And then the final thing is there's many sort of future questions or hypotheses. Does this apply to other protein sequences? So I've kind of, I focused on this one data set which looks at a particular protein family called P450, but there's many other protein families with different functionalities. And does this kind of apply to, does this general hypothesis apply to others as well. And if we use a GLM for other data sets, will it work? Will it work well? You know, can we combine multiple data sets together and get a similar conclusion? Like if, as I mentioned, there are multiple protein families, like what, like if we combine those families together, would we still get pretty similar conclusions to what we have here? And so this is just a couple of the many different questions that you can ask from a scientific point of view, and potentially develop methods that address those questions too. So there's lots and lots of future questions that you can ask. And once again, I kind of just expect you to think about this when you address this question on future directions. And I've just done a demo for this example just to be a little bit concrete. But for example, if you're thinking about voting, you could think about, well, how do you sample your data? I mean, one common question that people have thought about when they've talked about voting data is, why are the polls so poor at predicting the results? And so that's kind of, there's many different theories on that, and that's kind of, like maybe think about doing the sampling in a different way or something like that. Those are sorts of the kinds of questions I expect you to kind of be thinking about when you're addressing the sort of future questions or future directions part of this project. Okay, so that's kind of just that's basically everything that should hopefully set you up well to sort of know what's expected for the course project, and that'll be it in terms of the lecture material. We'll still be having office hours, so you can certainly sort of raise any questions that you have about the project during those office hours. And yeah, so good luck with the project, and we'll certainly be in touch. Okay, thanks. That's it for today and for the lecture material for the course.